Hello, I'm Emily Miyoshi. I am a senior bioinformatics scientist here at n-Lorem, and I just wanted to thank you for attending my presentation on how we're using omics data to inform and refine ASO design. So I want to first start off with what is even omics data. So datasets where you have the omics suffix is going to generally mean that all of whatever was being studied was quantified. And so, for example, genomics dataset, that means that we should have information for all the human genes. Transcriptomics, all transcripts or RNAs, and proteomics, all proteins. And high-throughput sequencing is a method where we can actually generate this omics data. And very simply, high-throughput sequencing is a method where we obtain millions of different sequences all at once. And so because researchers have made such great advancements in sequencing, the cost for sequencing has really dramatically decreased, and consequently, the number of omics datasets has really exploded. So now we have all this data. What can we do with all this data? And of course, advanced ASO discovery. However, not all data are equal. Unfortunately, there's just sometimes when you get datasets where the quality is just not very good. And so it's really critical that we identify well-annotated, high-quality, and standardized data that we can use to reliably inform the different steps of our ASO discovery workflow. And so in this presentation, I'm going to cover some of the different data that we use here at n-Lorem and provide examples of how we're using them to help us in the early stages of our ASO discovery. And I'll just briefly cover some of these different steps. So we first start with our feasibility assessment. Do we think we can successfully develop a therapeutic ASO? We actually have to ask this question for every gene, every mutation, for every patient really. And if we do think we can be successful, then we can move to the design stage where we want to design as many possible ASOs. And if we're really lucky, we can end up with thousands in this initial design stage. And if we do happen to be that lucky, then we will actually systematically filter or prioritize ASOs into a smaller set that we will then take into in vitro screening. And so for all three of these different steps, we use omics data in order to help us. So I want to first go through some of the large-scale public datasets that we're using to help guide our evaluation of feasibility as well as our ASO design strategy. And so the reference genome assembly, to begin with, this is a data set that a lot of different researchers are going to refer to because this is a consensus representation of all the different human genes. And specifically for each gene, we have the information of the different transcripts or different forms of RNA that are transcribed from said gene, as well as its sequence and the different sequences of these different transcripts. There's also a whole lot more information than just that. But you can get these data from sources like GenCode or Ensemble. I want to point out Ensemble in particular because it has a really nice web interface so that anyone can really explore this kind of data. But using the reference genome assembly, we're able to identify regions that we may want to exclude so that we can more efficiently design ASOs. And so some of these regions might be problematic sequences. So regions where you have very repetitive sequences, those are places there that we would like to avoid. In addition, there may be regions where the sequence is also found in another gene. And this could result in unintended effects or off-target effects, where our ASO could bind to our target gene as well as this other gene. And so we want to avoid this and then instead only design ASOs into these good regions. We also use population-level genomics data. So this is in addition to the patient-specific genomics data that we'll ask for. So we essentially want to also know what are the different genetic variants that have been identified in a larger cohort of people. So this could be globally or even in specific populations. And we're able to obtain this data from sources such as Genome AD. And we're essentially using these different genetic variants that have been identified in a larger cohort to help guide us in our ASO design strategy. For example, we'll use where the different genetic variants are located within a gene. This might help us to identify regions where we want to exclude. And this is really thinking about how can we treat more patients. Maybe we want to exclude where a variant would then make it so that this ASO would not work for another patient. We also look at the frequencies for these different variants. Is it commonly found in different people, or is it actually a really rare variant? So this goes again to this idea of how can we design ASOs that are going to be helpful to a larger number of patients. Another resource that we use is these atlases where we have the gene and protein expression information for a gene in multiple different tissues or different cell types. And some of the places that we get this information from are GTEx or the Human Protein Atlas. We use this information in order to help inform our feasibility and our safety assessments. So for example, is a gene expressed in multiple different cell types, or is it expressed in only one? This is going to help us to identify if we have actually a cell line that we can use for in vitro screening. And then for our safety assessments, depending on the way that an ASO is going to be delivered, we may see that we have a potential off-target effect with an ASO. However, if the off-target RNA is not actually expressed in the tissue that we are going to target, then in this case it may not be that much of a concern. And next, I want to go over how some of the technological advancements in sequencing have allowed for greater breadth and precision in our ASO discovery. And so high throughput sequencing, traditionally this is going to be referred to as next generation or short read sequencing. And what defines this as short read is that the sequences are going to be about 100 nucleotides in length. And when you think about the length of a gene, this can actually be more on the order of thousands. And so now we have long read sequencing. And this is really advantageous because instead of having maybe 100, you're getting more like 5,000 different nucleotides in a single read. And so one of the ways that long read sequencing has benefited us is in the design of our allele selective ASOs. So these are our ASOs where we want to selectively target and degrade the mutant RNA while not touching the wild type. And in order to do this, we need to identify, in addition to the pathogenic variant, we want to identify genetic variants that are also on the mutant allele or the mutant RNA. And this is how we'll be able to distinguish from our wild type. And in some cases, we may be lucky where there are variants very close to the pathogenic variant, that they are within that short read length. However, oftentimes they're not going to be that close, unfortunately. Instead, they could be very far away from your pathogenic variant. And so with short read sequencing, what we'll need to do is instead inference. We might need to use the information from parents or from even the population in order to kind of guess which variants are also on the same allele. However, with long read sequencing, because they can span such a large distance, the reads themselves are very informative and can tell us which variants are on that same allele as the pathogenic variant. And so in the cases of our allele selective ASOs, where we're trying to selectively degrade a mutant RNA, with long read sequencing, we can use just the patient's DNA sample, and this will give us direct evidence of whether or not a variant is on the same allele as the pathogenic. And so this gives us greater certainty for the variants that we're targeting, but also it can increase the number of variants that we can target. And ultimately, this gives us a higher chance of being able to develop a therapeutic. And so I also wanted to provide an example from some of our very own patients. So we have 4 n-Lorem patients with a pathogenic insertion deletion, or indel, in the gene GBE1. And what Dr. Orhan Akman back in 2015 had actually identified is that the insertion deletion will result in an exon that is not normally there in the GB1 transcript to be included in the mutant transcript. However, at that time, they had not identified how long the pseudoexon was, and they also believed that the exon was going to be the terminal or the last exon in the mutant GB1 transcript. However, back then, long-read sequencing really wasn't widely available, and it was also very expensive in order to perform this technique. And so when we were developing a therapeutic ASO for these patients, we actually performed long-read sequencing, and we found out that there's actually 2 different mutant GB1 transcripts, so 2 different lengths for that pseudoexon that's created from the pathogenic indel. And we also found out that the terminal exon in the mutant GB1 transcripts is not that pseudoexon. So that was another new finding where actually the terminal exon of the normal GB1 transcript is also included. And so in the case of these splice modulating ASOs, or ASOs where we're trying to force the wild type RNA to be created instead of the mutant RNA, it's really important that we have a complete picture of the different mutant transcript, or in this case, the 2 different mutant transcripts, because this ultimately is going to affect how we are designing our ASOs, or how we're developing our different screening assays, and also how we evaluate safety. In conclusion, through the careful selection and integration of these different omics datasets, we're able to rapidly assess feasibility. We're also able to more efficiently design ASOs while keeping safety in mind. And we're also able to develop ASOs that could potentially benefit more patients. We also are using new technology in order to advance our therapeutic development. I'd like to thank you for listening to my presentation, and I'd also like to thank our various sequencing providers and collaborators, as well as the many large research consortia who have generated and curated the public datasets that we use in-house. And so I hope you enjoyed the rest of your time at our colloquium. Thank you.